As I've mentioned in previous videos, the interaction between T cells and antigen-presenting cells with antigen is, for me, a love story. And when this occurs, remember, it occurs in a lymph node. So the T cells will have come in from the afferent lymphatic duct looking for their true love. The dendritic cell will have either migrated in through the lymphatic duct with an antigen, or will have found an antigen there that found its way into the lymph node. Either way, all three are in the same place, and now true love can meet. So we find the right antigen, then we get more signals that it's a good relationship through B7, CD28 interactions, and finally we get cytokines, which provide the differentiating signal to the T cell, basically informing the T cell what their life together is going to be like. All of this activity occurs within the T cell zone of the lymph node, which is the paracortex. At this point, our T cell needs to become a fully differentiated T cell and mediate its cell-mediated immunity functions. It can now travel out of the lymph node to the site of infection in order to carry out its effector function. An important note about this new effector T cell, you'll note that it left behind B7 and CD28. Now this is an effector T cell. Effector T cells no longer require co-stimulation. They've gotten all the signals they need to go out into the world and make something of themselves. That is true of both CD4 and CD8 positive T cells. And in this video, we're going to discuss the cell-mediated immune responses of CD4 positive T cells. This is kind of a two-part video. We'll discuss kind of the global consequences of creating one type of CD4 positive T cell, and that's the pro-inflammatory Th1 subtype of CD4 positive T helper cells. Then in another video, I discuss the other types of T helper T cells because there's a variety of subsets. Now, before we go too far afield, let's back up a little bit. So we're in the lymph node, and our naive T cell has received all the right signals. It's got CD4 binding to MHC class II, it's got the right antigen, it's got co-stimulation, heck, it even got some cytokines. Now, remember that this antigen-presenting cell could be a dendritic cell, as we've been discussing, or it could be a macrophage that has engulfed pathogen and isn't quite sure how to kill it yet. Or it could be a B cell that needs instruction on what kind of plasma cell it should differentiate into based on what types of antibodies it will produce. And this T cell now needs to give the antigen-presenting cell instruction. So one of the first things that will happen if we're talking about a Th1 CD4 positive T helper cell is that the CD4 positive T cell will begin producing a cytokine known as interferon gamma. We've talked about interferon gamma a lot, and this time interferon gamma is really going to get to shine. So interferon gamma gets produced by the T cell, and it acts on a macrophage that potentially is presenting antigen to our T cell. Interferon gamma was previously known as macrophage activating factor because it activates the macrophage, enabling it to kill the pathogens inside. And the way it does this is by inducing production of reactive oxidative products, increasing the production of lysozymes, and increasing production of reactive oxygen species and NO or INOS. The reason behind this is basically that it makes the macrophage a more effective killer. So by the CD4 positive T cell producing all of this interferon gamma, it's telling the macrophage, it's okay, kill that, kill what's inside you, and use all of this interferon gamma to create more toxic peptides and granules that will be able to kill the pathogen that you have engulfed. The other component involved in this macrophage activation is CD40 and CD40 ligand. So CD40, shown down here in green, is expressed on macrophages. And CD40 ligand will be upregulated on T cells following that third signal of differentiation during its priming. CD40 ligand also activates the macrophages in much the same way that interferon gamma does, basically instructing the macrophage to create more enzymes that are going to result in more efficient killing of engulfed pathogens. Now let's say for a minute that instead of a macrophage, our antigen-presenting cell is actually a B cell. B cells actually require T cell help for much of their effector function, which is to secrete antibodies. And unless the only type of antibody you're going to secrete is IgM, then you need T cell help, because T cells are the reasons that antibodies are able to class switch. And most typically, in the type of scenario we're talking about, a Th1 type T helper cell response will induce B cells to produce IgG antibodies. Now, class switching will only happen on the B cell if CD40 on the B cell is ligated by CD40 ligand on the T cell. So when the naive T cell becomes activated by the antigen-presenting B cell, it will be induced to upregulate CD40 ligand expression, which will then ligate CD40 on the B cell and induce the class switching events that result in the B cell developing into a plasma cell capable of secreting IgG. Now, once this T cell is activated, remember, it no longer needs co-stimulation. So this T cell is now able to, while it's still in the lymph node, go and interact with B cells within the lymph node. There, it can interact with them, bind to them, get stimulated by them again, and provide that CD40 ligand interaction to the B cell, which will promote the differentiation of this B cell into an IgG plasma secreting cell. And it can do that again with other B cells within the lymph node environment. Remember that plasma cells largely stay within the lymph node, so T cell help needs to remain in the lymph node. Remember also that these cells can leave the lymph node, and once they leave the lymph node, they can go and interact with macrophages and dendritic cells at the various sites in the absence of co-stimulation to promote the ability of antigen-presenting cells at the site of inflammation to kill pathogens that they have engulfed. So now that our CD4 positive T cell has been completely differentiated into a Th1 effector cell and has left the lymph node, it can get into all sorts of trouble. So it can activate macrophages to destroy bacteria through the production of interferon gamma and expression of CD40 ligand. It can kill chronically infected cells directly through expression of a molecule known as Fas ligand, which will literally cause the cell to kill itself when it ligates the receptor Fas that is expressed by the target cell. It induces proliferation of T cells because Th1 CD4 positive T cells produce a lot of IL-2, and everywhere they go, they're creating more IL-2, promoting more T cells to proliferate. And lastly, it can even promote chemotaxis of macrophages, and it does this in a couple of ways. It forces macrophages to differentiate in the bone marrow. It actually activates the epithelium, basically creating space in the wide junctions so that macrophages can actually arrive at the site. And they arrive at the site through production of CXCL12, which is also expressed by T helper cells, promoting their recruitment to the site of inflammation. So this goes into the effector functions of just one subtype of T helper cell. If you want to learn about the other T helper cell subsets, watch the other video looking at T helper cell subsets. The idea behind this is to really understand that once a naive T cell is activated in the lymph node, it becomes an effector cell. And this is basically entirely regulated by cytokine production in the environment by the antigen-presenting cells and other cells in the environment that basically license this cell to go out and attack cells that may have been infected or may hold pathogens within them and aren't quite sure how to kill them. The T helper cell is going to go out into the periphery and secrete cytokines or express receptors or create a situation where other effector cells through proliferation or chemotaxis can come and aid in destroying the pathogen at that site.